In this section, we'll have a general overview of adaptive immunity. Adaptive immunity detects and responds to particular microbial antigens from millions of possible other antigens. It's very good at detecting non-self. Adaptive immunity is known for its memory against previously encountered pathogens, and also known for its adaptive qualities in that it can adapt for each subsequent exposure. Adaptive immunity is also known for amplifying and focusing the innate inflammatory response. So as we mentioned in our innate lectures, with adaptive immunity we have more specialized cells dealing with pathogens that the innate system could not handle by itself. So the adaptive immunity kicks in, and with adaptive immunity we establish memory, and we also establish specificity for particular antigens and attune our bodies to be able to handle those specific infections with more precision and more effectively in later exposures. So here we see the entire spectrum of the immune response as it progresses, starting at the very early with the initial exposure of the microbe to the innate immunity. We see the mechanical barriers like the epithelial barriers. We see the phagocytes and the NK cells, the cellular component of innate immunity, and the non-cellular components such as complement, all taking place within those first 12 hours of being exposed to the microbe. Now, if adaptive immunity needs to kick in after that, we see that it start to come into play. The B lymphocytes start to be able to pick up on that native antigen, that antigen in its native form. We see the T lymphocytes starting to get exposure to their antigen through processing of the antigen-presenting cells. And all of this takes place over days, so that at the end of day 4, day 5, we start to see the initial primary responses of that adaptive immunity. But again, this is not an immediate response, in contrast to the innate immunity, which is very fast, very rapid. But this response, the adaptive response, is associated more with memory and specificity and specialized responses against particular antigens. So to break up adaptive immunity, there's two basic branches, the cell-mediated branch and the humoral branch. The cell-mediated branch deals mostly with intracellular pathogens. When you think of cell-mediated, you want to think of an antiviral type of response or some type of intracellular bacteria that you would need to be responding to. The major cell involved in the cell-mediated branch of adaptive immunity is the CD4 positive T cell, the helper T cell. Remember, this is our general. This is our cell that through cytokine release directs all other white blood cells to kill intracellular microbes, to get activated, to enhance phagocytosis. All of that comes through these T helper cells. CD8 T cells are also involved in cell-mediated immunity, but more through direct cytotoxicity. They go looking for cells that are altered, that are virally infected, or tumor cells that need to be destroyed. These cells in the adaptive or in the cell-mediated branch of immunity differentiate into effector cells, memory cells, and also regulatory cells, which we'll talk about a little later. The other branch of adaptive immunity is the humoral branch. This branch deals more with extracellular pathogens, parasites, and also bacteria that are more extracellular. And this basically is composed of B cells. So when you think humoral immunity, think B cells. And these B cells can be either thought of as T-dependent or T-independent. By and large, the majority of B cells are T-dependent. They produce antibodies with CD4 T cell help. There is a subset that is T-independent. This is a small subset that can produce antibodies in response to certain types of antigens, and we'll talk about that a little later as well. These cells can differentiate again into memory cells, just like the T cells, or plasma cells. And plasma cells are basically antibody factories. They upregulate their endoplasmic reticulum. They stop worrying about finding an antigen and solely concentrate on pumping out as much antibody as they can. So kind of an overview of the entire adaptive response goes something like this. The antigen is recognized by a specific naive lymphocyte in the lymph node, either recognized entirely by a B cell, which can recognize it without any help, or recognized through that MHC process with the antigen-presenting cells if it's a T cell. These cells will then differentiate into effector cells, and they will proliferate in what we call the expansion phase. The immune system is all about expanding and get as many cells out as you can, especially in this phase, specific cells. After the expansion phase comes the elimination of the source of antigen, be it through antibodies or through cytokines activating other types of cells. After the effector phase has gone through, these effector cells will die off. The plasma cells will die off. They won't live that long, and you will return to what we call the homeostasis, or the point of regularity for the amount of T cells your body, or B cells that your body is supposed to have. And we call this the contraction phase. At the end of the contraction phase, what we're left with is memory cells. And these memory cells are important in immunity because they evoke faster and more intense responses upon re-exposure to the same antigen. And we'll talk about that later as well. Some vocabulary that you need to be familiar with. A lymphocyte is a B cell or a T cell when we're talking about adaptive immunity. Remember, it's important to note that NK cells are also defined as lymphocytes because of their lineage, but they're not antigen specific. An immunogen is a molecule that generates an immune response. So any molecule that will cause your body to have an immune response to it is an immunogen. There are different factors that can enhance immunogenicity or make a molecule more immunogenic. These factors include foreignness, which would be how different this protein is compared to the proteins that are in your body. Molecular weight and size, with a high molecular weight and size being more conducive to immunogenicity. Its chemical complexity, and also its route of administration, parenteral versus oral. How did this immunogen get into your body? So an antigen, in contrast, is an originally described substance that generates antibodies. That's how it got its name. Now it describes any molecule that sensitizes specific lymphocytes. So anything that could cause an immune response is an antigen. However, going back to the previous slide, not all antigens are immunogens. So that's something to keep in mind. Anything in your body that could sensitize a specific lymphocyte would be considered an antigen. All the proteins in your body would be considered antigens. However, they're not all immunogens. Why? Because they're self-proteins. They're not foreign. They don't have the right makeup to be immunogenic. So that's something to remember and distinguish between antigen and immunogen. Anything that can cause an immune response is antigenic. Anything that does cause an immune response is immunogenic. Now, an epitope, to get a little bit more specific, is the antigen-determining site on an immunogenic molecule that lymphocytes recognize. So if you think about a large antigen, there's lots of different places where an antibody may be able to bind to that antigen, right? Each of those places would be considered an epitope. So you can have antibodies to multiple epitopes within the same antigen. So again, a little bit more specific. And if we're being technical, we would say that antibodies are epitope specific, not technically antigen specific. Hapten is another term that you need to know. Hapten is a small molecule that isn't antigenic by itself, but will become so when coupled to a larger carrier molecule, such as albumin or another host protein. So these are molecules that are smaller. Remember, we said that size had an effect in determining immunogenicity. So with a hapten, you have a molecule that's very small, so small that the immune system kind of ignores it. But if that molecule can bind to something that's a little bigger, say albumin, which is abundant in your blood, then all of a sudden the immune system can recognize it in the context of something bigger and can respond to it. So here we have a question. A pharmaceutical company is developing a drug to counteract the effects of allergic reaction to penicillin. They notice that when penicillin alone is incubated in the presence of purified peripheral blood mononuclear cells, it causes no immune reaction. However, when the drug is put into a whole blood culture, it will elicit an immune response. Which of the following best describes this molecule? A, immunogen, B, antigen, C, hapten, D, anti-inflammatory, or E, steroid. The answer here is C, hapten. What we're seeing in this question, and just in terms of vocabulary, anytime you see peripheral blood mononuclear cells, they're talking about white blood cells. So we see in the purified white blood cells, there's no immune reaction. But when you put it in with whole blood, you get a response. Well, what's in whole blood? These additional proteins, these carrier proteins. And so when you see something like this, this is a hapten. This is a haptenogenic effect. So different types of antigens that your body is exposed to. We can kind of break this down into exogenous as well as endogenous antigens. So exogenous antigens are what you typically think of when you think about an immune response, something from a bacteria, something in its cell wall, in its flagella, in its capsule, maybe an exotoxin, things that are foreign, come from a bacteria. Also viral, capsid proteins, surface receptors, nucleoproteins, things that your body will respond to, things that you expect your body to respond to in order to fight off these sicknesses. Now, another group of antigens is the endogenous antigens, and we see these more typically when we talk about hypersensitivities and autoimmunity, transplant rejection, things like that. So blood group antigens, like the A and B antigens or the Rh antigens, those are endogenous type of antigens that are produced not from something foreign but from something self that you could have a reaction to. Also MHC or HLA, just to distinguish between those two. MHC and HLA, if you see it on the boards, they're talking about the same thing. HLA stands for human leukocyte antigen. It's the MHC that's found in humans. So HLA is specific human MHC, versus if you just see MHC, they could be referring to MHC from any species, such as a mouse or a rabbit or whatever. So with antigen binding and recognition, the B cells use a B cell receptor. This is a membrane-bound antibody on B cells that has the same specificity as the antibody that particular B cell produces. When we talk about the genetics of B cells, you'll see how this plays in a little bit more. But B cells, remember that I said that they recognize their antigens as they exist in nature. And it's actually an antibody that sits on the surface of the B cell that provides the B cell receptor that is out in what the B cell uses to attach to its antigen. It will then endocytose it and it can break it down. For the T cell receptor, these are polypeptide chains that are anchored to T cells. It contains a groove which binds small peptides presented by MHC that is present on the APCs. So T cell receptors, remember, they have to have antigen processed. They're a little bit pickier. They need something broken down and presented to them. Now, what's MHC? Major histocompatibility molecules. These are present on all nucleated cells, and it's essential for peptide presentation to T cells. Remember, in earlier sections we talked about what cells express MHC class 1 and what cells express MHC class 2. All nucleated cells present class 1. Antigen presenting cells, that is, macrophages, dendritic cells, and B cells, express class 2. This concludes our discussion of MHC. In the next section, we will discuss humoral immunity.